concept. So mechanisms of vaccine induced immunity. So as I told you, we have different kind of APCs in our body. So you cannot imagine that your immune system will work in a very satisfactory manner if you are having the impaired APCs. Like if you are not having the well functioning APCs, then most of the things will not be communicated within the immune cells. Like there are different immune cells in our body. So they are responsible to provide the immunological response against whom? Against the antigen. So they do not do this alone. They need the association of different cells like APC. So the foremost role in immunology is for in the context of immunological response I am talking. So that APCs are the first which are recruited to the site where they recognize that yes, we have encountered an antigen. We have to process it along with whom? I taught yesterday, along with MHCs. So along with MHCs and APCs are the major, you can say, pillars of our immune system. If APCs are not working properly, then your body will not be able to provide the immune response. Why is it so? Because the antigen is not antigen will not be processed and will not be presented to the immune cell. यानी उसे परोस के ही नहीं दिया जाएगा. So immune cells are alone not at all functional to recognize the antigen. They have a function to provide the immune response once it is recognized by the APCs along with the MHCs. Now, so the first role is the what vaccines do. So vaccines mimic the pathogen, as we have studied about the role of adjuvant also, and also we have studied the role major mechanism of how the vaccines work. So vaccine mimics the pathogen because what we are taking in the vaccine, we are taking nothing but the antigen, which is attenuated, which is inactivated, which is killed, which is not that much lethal when it was in its earlier stage. So in the form of vaccine, we are providing antigen in our body. So the vaccine mimics the natural antigen condition. Now, whenever we are taking vaccine, our immune response will consider the antigen as a non-self, and then it will start producing different kind of immune response. So you can have the, as I told you so many times in my previous lectures, that first line of defense is always our innate immunity. For the very first time, not at all adaptive response will be activated. For the very first time, we will be having the innate immune response, which is followed by the adaptive immunity, which is, you can say that memory, immunological memory. So this one is an important part. So how the efficacy of a vaccine is measured? Like if you are saying we are having two types of vaccine, vaccine 1 and vaccine 2 for let's say HPV. So how you can say that V1 is effective or V2 is effective? So you have to say that you have to analyze the whether V1 is capable of inducing the immunological memory or V2. So if V1 is not able to induce the immunological memory and this V2 is capable of inducing the immunological memory, then you can say that yes, V2 is more efficient against HPV as compared to the V1. So let me explain why this is so. This is because whenever your body will having memory, so it is not at all, you cannot say that if the antigen has entered your body for the first time, it will not enter for the second, for the rest of the, or you can say for the subsequent times, it can enter your body. You cannot say that it will only enter for the very first time. So for recurring events, we need memory. If our immune system will have the memory that HPV to pehle bhi aaya tha, so it will recognize it very quickly and accordingly it will react for the second time, for the third time, and Therefore, the severity of the infection will be increased or decreased? Decreased. It will be very much decreased. So the effectiveness of the vaccines are measured in terms of how much immunological memory they will boost inside the host, or you can say how much antibodies, because it memory can be measured in terms of antibodies. So in case of COVID-19 vaccines also, the blood microtiter values are recorded, that whether the antibodies are increased or not. So if the patient having increased antibody, let's say Covaxin has increased the antibody titer of a particular patient, then you can say that it has a adaptive immunity, immunological memory. So whenever the COVID-19 will attack the same patient for the second time, then the severity or the infection intensity will be much, much lower as compared to the former one. So this is the concept of how the vaccines boost our immune system. So three things will be cleared, that first, the vaccine mimics the pathogen. Second thing, that it can uh, boost our innate immunity, but for the very first time your innate immunity will be boosted, and after that, after subsequent exposure, you will be having immunological memory, that is more or less the adaptive immunity. Now we are having T cells. So APCs present the antigen to T cells, that is CD4 helper T cells and CD8 cytotoxic T cells. So yesterday I told you, and in my so many videos, that MHC1 and MHC2. So we are having this APC. So APC will work with this MHC1 and MHC2. So MHC1 is for TC cell, MHC2 is for T helper cell. So they present the what? They present the processed antigen. It means, suppose this is an antigen, bacteria. So they will cleave it out, and the fragment will be, this is T cell receptor. They will present it to the T cell, and how they will present it? There must be some receptor. So that is known as T cell receptor. TCR are a very important part of immunological syllabus. So they present it to the T cell, and then T cell will behave accordingly as per what antigen it is. So we are having two different types of T cells, and the helper T cell will do what? It will activate B cells, and it will produce antibodies like IgG, IgA, and a lot more. We are having GAMDE. You can remember 5 types of antibodies we are having: IgG, IgA, IgM, IgD, IgE. So you can remember this like game, and you only have to put D. So we are having 5 types of antibodies in our body. So see, T cell antibody नहीं बनाता है, B cell antibody बनाता है. तो T cell, B cell को antibody बनाने के लिए help कौन करेगा? T cell will help B cell to produce more and more antibodies. यानी प्लाज्मा सेल, बी सेल भी डिवाइड होता है प्लाज्मा एंड इफेक्टर सेल में, और वो प्लाज्मा सेल ही है जो एंटीबॉडीज रैपिडली बनाता है. एट हाई टाइटर्स वो बनाता है, जैसे वो एंटीजन लोड बढ़ते जाएगा बॉडी में, इट विल स्टार्ट सिंथेसाइजिंग एंटीबॉडी रैपिडली. So some B and T cells become memory cells for long-term protection. Adjuvant enhance immunogenicity by boosting innate immune signals. So ये जो 4-5 लाइन मैंने अभी लिखी है, उसके लिए मैंने इतना जो समझाया है, this is the summary of how the vaccine induce our immunity. So 3-4 concept यहीं से immunology के पकड़ लो कि you have to study about the different kind of APCs. Secondly, you have to study about the different types of T cells, and majorly आपको क्या पढ़ना है कि B cell is involved in the antibody production. That is, we are having 5 types of antibodies, and T cell और B cell दोनों ही lymphocytes हैं, but B cell produces antibodies along with the T cell, like not along with the T cell. in association with T cell, you can say. T cell help B cell to produce the antibodies. Now move further. So see, this is a beautiful diagram of cellular immune response. Like if we are administering any such nucleic acid vaccine like DNA vaccine. Nucleic acid vaccine means mRNA vaccine can also be there, RNA vaccine can also be there, recombinant DNA vaccine can also be there, a lot more thing. So basically this is nucleic acid antigen, which is specifically DNA vaccine. So if you are administering a patient with DNA vaccine, what will happen? Transfection of APCs. Like APC में transfection होगा. Transfection means the APCs will take up the DNA. So transfection शब्द का मतलब होता है कि suppose आपने कोई भी recombinant कुछ बनाया है और उसको host में आप एंटर करा रहे हो. सो ये अगर इसमें आ गया, देन यू कैन से दैट दिस सेल इज ट्रांसफेक्टेड. ट्रांसफेक्शन से ये मत सोचना कि इन्फेक्शन. ट्रांसफेक्शन मींस ट्रांसफर हो चुका है जो भी सब्सटेंस था. इट इज नॉट ओनली हमेशा इन्फेक्शन ही होगा. सो ट्रांसफेक्शन होगा एपीसीज का. एपीसी कौन कौन से हैं? वी आर हैविंग बी सेल्स, dendritic cell and macrophages. So these are the APCs in our body. So सबसे पहले जैसे ही हम दे रहे हैं DNA vaccine, then you can say antigen gene will be taken up by the APCs, right? उसके बाद क्या होगा? As I told you, APC never function alone. It will work along with the association of MHC. So you can see here it is coordinating with the MHC class 2, like it is the processed form of whatever DNA vaccine antigen, like antigen gene is processed, it is hydrolyzed, and along with the MHC, it will be presented to the T cell receptor. And you can see MHC class 2 and here MHC class 1, because for TC cell and for T helper cell, because we are having two variants of T cell. So MHC 2 will work for TH cell. Don't be confused. And ये H से समझ जाना कि ये क्या है, T helper cell, and ये 2H, मतलब मैं अपना ऐसे याद रखती थी, ये ऐसा है मतलब ये H जैसा दिख रहा है. So TH is for MHC 2 and for MHC 1 TC cell, right? So you can see they are in association with MHC class 1 and class 2. They will present it to the TCR, and then TCR will do what? It can react through the humoral immune response. Humoral immune response means blood mediated. So this can also be true, or else they can be easily turned to apoptosis. There are different kind of crosstalks going on inside our body. You cannot say that only a single mechanism will take place. No. Suppose if you are having like 100 genes from the DNA vaccine, because if you are taking, let's say, a single dose for an explanation purpose, for just to make you explain in an easy way, I have considered this 100. Suppose 100 genes are going inside your body, and suppose APCs are taking up the 100 genes. So it is not necessary that the 100 genes will only follow this process. Some 10 will follow this process, some 10 will follow that process, some 10 will follow the, not only just 10, 2, 3, 2, the approximate percentage will be different in different host. So there are so many crosstalks going inside our body. So this is the drawback of each and every therapeutic drug. Like if we are targeting any particular, like in cancer also, why cancer is very difficult to treat? Because whenever we will target the pathway A to block the cell proliferation, the crosstalk according to the different mechanism, you will say that the pathway B is stimulated or either inhibited by whatever you have given to the pathway A. So this is known as crosstalks, when so many pathways are going on simultaneously. So you can have different cytokines released, and these are the inflammatory cytokines, and you can say that it will be uh, increasing, it will increase your immunogenicity. Now you can see these apoptotic cells. So some of the antigens are directly turned to the apoptotic pathways, and these are the antigen. You can say that these are processed, these are hydrolyzed, and they are presented to the T cells to make B cell produce antibodies. So this is how your overall cellular immune response works. Cellular means cell mediated. How many cells are involved? APCs are involved, and also apoptotic cells are involved. In B cells are involved in producing human antibody. T cells are involved, dendritic cells are involved, like APCs are involved mainly. So it is cell mediated. You can say myocyte, keratinocyte, B cell. CTL means cytotoxic T lymphocyte. CD4, CD8. So you can say that we have to take a lot of care while doing the transfection of a particular vaccine into the host. It means whenever you are delivering or administering a particular vaccine in a patient, you have to take care that what pathway will be targeted by this vaccine, because we have to take care of this crosstalk. Like it should not be that it is doing well in the pathway 1, but it is due to the JAK inhibition mechanism is also going on. So this is the overall mechanism. Now this one is another example, like if you are taking edible vaccine, like oral route से अगर हम कोई vaccine ले रहे हैं, so what it will be? It will be, you can say that we are having antigen from the edible vaccine. सो एडिबल वैक्सीन से जब भी डायरेक्टली हमारे पास एंटीजन आता है, सो इट विल, वी आर हैविंग मैक्रोफेजेस इन आवर बॉडी. So macrophages, as I told you, is also a APC. 3 types of APCs are there. B cell, always remember, macrophages and DC cell. DC means dendritic cells. So macrophages are also the APCs. So they will just take up the antigen from the oral vaccine, whatever you have taken edible vaccine, and from that it will, you can see it is this I was talking about. Just a minute. This I was talking about, this structure. This is known as TCR. You can see this is T helper cell. So always the APCs will present the antigen to whom? To T cell, because T cell will direct the B cell कि अब एंटीबॉडी बनाने का समय आ गया है. सो बी सेल अपने आप से नहीं बना पाएगा, उसको सिग्नल कौन देगा? टी सेल. सो यू हैव टू रिमेंबर दैट जब भी एंटीजन आएगा उसको कौन सबसे पहले सेंस करेगा? एपीसी करेगा. एपीसी किसके साथ में मिलके करेगा? एमएचसी के साथ में मिलके करेगा. एंड उसके बाद क्या होगा? दे विल प्रेजेंट इट टू टी सेल रिसेप्टर, यानी मान के चलो टीएच और टीसी सेल, एंड आफ्टर दैट बी सेल फंक्शन विल कम. सो सी, टी हेल्पर सेल ने किसे डायरेक्ट किया? बी सेल को बोला कि अब हमारे बॉडी में एंटीजन आ गया है, नाउ इट्स टाइम टू प्रोड्यूस द एंटीबॉडीज. नाउ दिस इज द एक्टिवेटेड बी सेल, ये फटाफट एंटीबॉडी बनाने लग जाएगा. वाई शेप, वी आर हैविंग वाई शेप एंटीबॉडीज. सो ये फटाक से क्या करेगा? एंटीबॉडीज बनाने लग जाएगा. सो दिस इज द वे अ वैक्सीन वर्क. यानी जरूरी नहीं है कि सिर्फ एक ही पाथवे सबके लिए एप्लीकेबल है. Now in this also you can see this is a viral antigen. Suppose you have taken a vaccine which is targeting any such viral disease, any कोई virus को target कर रहा है. So you can see APC सबसे पहले क्या करेगा? Along with MHC2 and MHC1, because it has a site for the binding of both MHC1 and MHC2, because you have to offer the antigen to TC cell and T helper cell, not only just TC and सब दोनों को ही देना है. TC and T helper दोनों को देना है. So it must be MHC2 and MHC1. So दोनों देखो यहाँ पे दिख रहा है. And what it will do, वो TH cell को देगा, and then TH cell will do what? वो साइटो टी लिम्फोसाइट को, साइटोटॉक्सिक टी लिम्फोसाइट को एक्टिवेट करेगा. दे विल सिंथेसाइज इंटरफेरॉन गामा. इंटरफेरॉन इज फॉर दी वायरस को, वायरस इन्फेक्टेड सेल्स को किल करने के लिए रिलीज होता है. परफोरिन ग्रेन्जाइम यानी ग्रैन्युलर एंजाइम रिलीज करता है. ये सब कौन रिलीज करता है? टी सेल रिलीज करता है. परफोरिन मीन्स इट विल स्टार्ट मेकिंग पोर्स ऑन दी एंटीजन. ग्रेन्जाइम मीन्स ग्रैन्युलर एंजाइम्स रिलीज करेगा, जिसकी वजह से ये पूरा जो फेगोसाइटोसिस इंक्रीज होगा. एंड ऑल्सो इट विल लीड टू मेमोरी टी सेल. And B cell को भी activate कर रहा है to produce antibodies. Now, as I have written here, one point, if you remember, some B and T cell become memory cell for long-term protection. So you can see here in this chart, whenever this TH cell is dividing into cytotoxic T lymphocyte, and also one cell is converting into memory T cell for storage, and in B cell also one is producing antibody and one is converting into memory B cell. Now this will be stored for the second response. So this is the way how vaccines work. Now the I just explained through the diagram, like these are the things written, like ये notes है जो मैं अभी तक समझा रही थी diagram के through. Now see, vaccines stimulate the body's immune system to recognize and combat pathogen. So pathogen can be of different type. जरूरी नहीं कि वो सिर्फ वायरस ही हो. इट कैन बी बैक्टीरिया, इट कैन बी फंजाई, इट कैन बी हेलमिंथ, इट कैन बी एनीथिंग. सो इनेट इम्यून रिस्पॉन्स इज इमीडिएट एंड नॉन स्पेसिफिक. ये सारी फैक्चुअल्स में आता है CSIR के B सेक्शन में. यही सब अच्छे से याद नहीं रहता है तो हम सॉल्व नहीं कर पाते. सो इनेट इम्यून रिस्पॉन्स इज क्विक एंड यू कैन जस्ट फिगर आउट. कोई भी चीज क्विक होगी तो क्या होता है? सपोज हमारी भी किसी से लड़ाई होती है, जब हम एकदम ओवर रिएक्ट करते हैं, क्विकली ऑन द स्पॉट अगर बोल देते हैं बिना सोचे समझे, तो क्या होता है? बाद में रिग्रेट होता है. अगर हम वही बहुत सोच समझ के करते हैं तो चीजें अच्छी होती है. लाइक इफ यू आर अटेम्प्टिंग एनी सच मल्टीपल चॉइस क्वेश्चन, सो विदाउट थिंकिंग, इफ यू वांट टू attempt it very quickly to save your time, then there are chances that you may have some incorrect attempt. But if you are properly taking time, properly reading the question, and then you are answering it, then it will be delayed, but the chances of that the question will be correct is more increased. So समझ आया difference innate और adaptive का? Innate तुरंत response देता है, so therefore we are having this नॉन स्पेसिफिक, यानी कभी कभी फास्ट रिस्पॉन्स देने के चक्कर में नॉन स्पेसिफिक रिस्पॉन्सेज हम दे देते हैं थ्रू द इनेट इम्यून रिस्पॉन्स. बट वी आर हैविंग द डिलेड, यानी देर से रिस्पॉन्स आएगा बट जो रिस्पॉन्स आएगा वो सही आएगा. सो फर्स्ट लाइन ऑफ डिफेंस, एंड दिस इज the later on response which we generate through the first exposure. यानी इसका मतलब ये नहीं है कि अगर innate नॉन स्पेसिफिक है तो ये किसी काम का नहीं है, बिल्कुल भी नहीं. We are saying that innate immune response अगर नहीं होगा सो ये भी नहीं होगा, because memory तो तभी develop होगी ना जब वो first encounter करेगा pathogen. So always innate immune response is the major part of our immune system. So first line of defense which are triggered by the PMPs. Now you have to remember PMPs are part of antigen. ये antigen के ऊपर जो molecules होते हैं जो recognize करता है हमारा APC, उसे बोलते हैं PAMPs. कई बार मैं बता चुकी हूँ, पैथोजन एसोसिएटेड मॉलिक्यूलर पैटर्न्स. APC एंटीजन प्रेजेंटिंग सेल. एक जगह इसका फुल फॉर्म लिख लिया करो जहां पे भी आ रहा है, एंड हमेशा फुल फॉर्म लिखने की जरूरत नहीं है, शॉर्ट फॉर्म में लिखा करो. लेकिन ये नहीं कि भूल गए उसका मतलब. PMPs किसी ने पूछा तो पन्ना पलटना पड़ रहा है, नहीं. So antigen presenting cell like dendritic cell and macrophages will recognize vaccine antigen. They will process and present them to MHC molecules, and what they will do, MHC molecule क्या करता है? See here, they will release cytokines. Cytokines release करेगा which will be inflammatory responses. So these are the thing. What kind of cytokines they can release? Interleukin 1, interleukin 6, tumor necrosis factor alpha, and different kind of interferons. It depends on what type of antigen it is. अगर viral होगा तो अलग-अलग type के release होंगे, bacterial होगा तो अलग-अलग type के release होंगे. Now adaptive immune response. So we are having delayed and specific kind of response. which is generated after the repeated exposure of the first antigen. So it is initiated by the interaction between APCs and T lymphocytes in the lymph nodes. T cell activation कैसे होता है? CD4 plus T helper cells like TH1 and TH2. बहुत बार हमने TH1 TH2 पढ़ा है. So TH1 what it does, it activates the macrophages and CTL. So CTL are nothing but the cytotoxic T lymphocyte, which are cell-mediated immunity, and it stimulates B cells to produce the antibody. So this is humoral immunity. हमेशा याद रखना कि जब भी टी सेल का इन्वॉल्वमेंट होता है हमारी इम्यूनोलॉजी में, सो इट इज नोन एज सेल मीडिएटेड इम्यूनिटी. But if your B cell is involved, so it is always considered as a part of our bloodstream. And humoral means what? Blood में ही होता है, हम वो directly हमारे blood में बात होता है. So it is part of the humoral immunity. तो simply कुछ terms key points है, अगर वो समझ लिया तो immunology is very very easy. अगर बेसिक्स समझ लिया तो जब भी बी सेल की बात होगी तो किसकी बात हो रही है? ह्यूमोरल की. अगर किसी को याद होगा तो इनिशियल क्लासेस जो मैंने डिफरेंस लिया था ह्यूमोरल इम्यून रिस्पॉन्स एंड बी सेल का, वो इसीलिए लिया था ताकि आगे जब हम पढ़ेंगे तो हमारा बेसिक क्लियर रहे. टी सेल की बात होगी तो हमेशा सीएमआई की बात होगी, सेल मीडिएटेड immunity. Now you have to always remember this TH1 and TH2. These are not just the different kind of T helper cell, but TH1 is always involved in the CTL, cell mediated immunity. They will activate the macrophages. Macrophages क्या करता है? It will do the phagocytosis. Phagocytosis में it will engulf the antigen rapidly. और यही जब रैपिड एनगल्फिंग होता है, रैपिड फेगोसाइटोसिस होता है बहुत ज्यादा लार्ज अमाउंट में, सो इट इज नोन एज ऑप्सोनाइजेशन. Opsonization is nothing but increased phagocytosis of the antigen, and TH2 will always stimulate the B cells to produce the antibodies via the humoral immunity. Now CD8 plus, that is cytotoxic T lymphocyte. As I told you, CD8 हमेशा MHC1 ही recognize करता है, and जो MHC2 है, MHC2 will recognize what? T helper cell. So you have to remember all these things: CD8, CD4, MHC1, MHC2, TH1, TH2. So I don't think it is difficult because यहाँ पे सारे pointers mentioned हैं. You can just take the screenshot or else I will share it after the completion of 6th unit. तो 6th unit तक के notes आपके पास रहेंगे so that you can revise it. Now B cell activation. So upon recognizing antigen and receiving help from TH2 cells, B cell undergo clonal expansion and differentiation. Plasma cell and memory B cell. So you have to just remember कि B cell में भी CSIR question जब पूछता है तो वो specific questions पूछता है B section में to test your factual knowledge. So B cell सबको पता है कि T और B में से कौन बनाता है? B cell बनाता है antibodies. But अगर और specific पूछे तो plasma cell, memory B cell. So it is the plasma cell which synthesizes the antibodies, and memory B cell is stored for the long-term immune system, long-term immune response. Now, we cannot forget the role of adjuvants. So adjuvants are involved in increasing the immune response by increasing the immunogenicity of whatever antigen we are taking. So as we know, these are some of the commonly used adjuvants. Now, I think यहाँ तक अच्छे से crystal clear है. अब थोड़ा सा आगे बढ़ते हैं. So second part of the 6th unit, what it is, immune response to different types of vaccines. So I have also included the detailed part. So firstly discuss this one very quickly. So this is the first part. We are having different kind of vaccines: live attenuated vaccine, inactivated killed vaccine, subunit, toxoid, mRNA, viral. So we have already studied about the vaccine type. So you can revise it if you want to just clear your understanding of this topic. So what kind of immune response is elicited by this live attenuated vaccine. So it can lead to strong cellular and humoral response, and a lifelong immunity is possible. And inactivated killed vaccine will increase what? प्राइमरी ह्यूमोरल. सो इट रिक्वायर्स बूस्टर डोज. सो अगर आपको पिछला वाला याद है, यानी अलग अलग टाइप के वैक्सीन को किसके में बूस्टर डोज की जरूरत पड़ती है, किसका रिस्पॉन्स कितना है, सो ये वाला जो पार्ट है वो आपको अच्छे से समझ में आ जाएगा अभी. सो वी एज वी ऑलरेडी नो कि हम लोग अगर लाइव एटेन्यूएटेड ले रहे थे, सो क्या हो रहा था, लाइव एटेन्यूएटेड ले रहे थे तो we are just decreasing the what to say the viral load को हम कम कर रहे थे, not viral load, एंटीजेनिसिटी हम कम कर रहे थे. किसकी? वायरस की. यानी इसमें हम मारते नहीं है, जैसे इसमें इनएक्टिवेट किल करते हैं, इसमें हम बस उसका जो वायरल प्रॉपर्टी है वो हम थोड़ा सा डिक्रीज कर देते, थोड़ा सा दबा देते, ताकि वो बहुत ज्यादा ट्रिगर ना करे इम्यूनोलॉजिकल रिस्पॉन्स. सो यहाँ पर क्या हो रहा है, लाइव एटिन्यूएटेड में वो स्ट्रॉन्ग सेल्यूलर और ह्यूमोरल रिस्पॉन्स देगा, क्योंकि वो वायरल पूरी तरह से किल्ड नहीं है, वो वायरस वो अभी एक्टिव है. नाउ इन दी सेकेंड इफ वी आर किलिंग इट, सो ये उतना स्ट्रॉन्ग रिस्पॉन्स नहीं देता है, एंड देयरफॉर इट रिक्वायर्स बूस्टर डोज. सब यूनिट में क्या करता है, ह्यूमोरल रिस्पॉन्स ही जनरेट करेगा, यानी एंटीबॉडीज ह्यूमोरल आ रहे हैं, मतलब बी सेल एक्टिवेट होगा और बी सेल एंटीबॉडी बनाएगा. इट इज सेफर बट नीड्स एडजुवेंट टू इंक्रीज दी, because we are taking only a subunit, only a part like protein, carbohydrate, antigen का एक part ले रहे हो. Toxoid produced antibodies against the toxins like DBT vaccine, mRNA like COVID वाला जो vaccine हमने लिया. Now come back to this one. This is detailed one. So each vaccine type triggers a different pattern of immune response depending on how closely it mimics the natural infection. So as I told you, these are the different categories of vaccine which we administer in the patient. So you attenuated means weakened pathogen that replicates minimally. So they can lead to cell-mediated and humoral and as well as long-lasting. So what are the examples? MMR, BCG, oral polio, yellow fever, etc. So these are under the category of live attenuated. And in the inactivated one, in which we kill the antigen, so therefore अगर वो kill हो गया है तो response कम ही देगा ना, intensity तो कम ही होगा. So therefore booster dose are required, and inactivated poliovirus and rabies, hepatitis A, all those are example of inactivated and killed. Now you can see subunit में क्या example है, hepatitis B, HPV, acellular pertussis, जो whooping cough होता है वो. टॉक्साइड में टेटनस, डिप्थीरिया. सो यू कैन सी mRNA का अभी रिसेंट एग्जांपल आपको याद होगा, फाइजर, बायोटेक एंड मॉडर्ना. सो ये जितने भी है वायरल वेक्टर, जिसमें एडिनोवायरस हमने यूज किया था कोविशील्ड के लिए, मोस्ट ऑफ द लोगों को कोविशील्ड ही लगा था इंडिया में. सो वी आर हैविंग different types of immune response as per the vaccine type. So you can see viral vector लिया है तो potent cell mediated और humoral response generate हो रहा है. mRNA vaccine लिया है so antibody response भी आ रहा है, and यहाँ पे T cell का भी involvement है along with the APCs. Conjugate में क्या होता है, इसका immune response generally ये immune किसको दिया जाता है, infants and children को दिया जाता है. So यू कैन सी ब्रेन रिलेटेड जो मेनिंगोकोकल वैक्सीन होते हैं, यानी मेनिंग्स में जो डिजीज होते हैं हमारे ब्रेन के कवरिंग में, उसके लिए ये दिया जाता है. सो इट विल लीड टू सम डिफरेंट काइंड ऑफ रिस्पॉन्स. सो इट डिपेंड्स ऑन दी यू कैन से दी इंटेंसिटी ऑफ द एंटीजन. अगर वो एंटीजन बहुत ज्यादा, यू कैन जस्ट क्विकली राइट इट डाउन इन योर नोटबुक कि अगर एंटीजन बहुत पोटेंट है, बहुत स्ट्रांग है, तो बहुत ज्यादा इम्यून रिस्पॉन्स वो जनरेट करेगा. एंड कभी भी अगर एंटीजन स्ट्रांग होता है तो हमारे इम्यून सिस्टम का जो दोनों पिलर है, व्हाट आर द पिलर्स, वी आर हैविंग सीएमआई एंड ह्यूमोरल, तो वो दोनों ही एक्टिवेट होगा, बिकॉज एक से काम नहीं चलेगा. सो बोथ विल बी एक्टिवेटेड. सो इस तरह से कॉन्सेप्ट को यू कैन मेमोराइज कि अगर दोनों ही है हमारे में, अगर वो एंटीजन बहुत स्ट्रांग है, एक से रिस्पॉन्स से वो किल नहीं हो सकता, वो इन्फेक्शन का जो लोड है वो बढ़ा देगा बॉडी में. सो बोथ आवर पिलर्स विल बी एक्टिवेटेड, यानी दोनों ही मिलके काम करेंगे. बट ऐसा है कि नहीं, ये तो ऐसे ही छोटा मोटा कच्चा सा है, तो क्या होगा, सिर्फ ह्यूमोरल ही संभाल लेगा उसको. इट मीन्स सिर्फ एंटीबॉडीज प्रोड्यूस होंगी एंड इट विल बी इरेडिकेटेड आउट. सो इट डिपेंड्स ऑन हाउ स्ट्रॉन्ग एंड पोटेंट द एंटीजन इज. तो कभी भी क्वेश्चन आए तो हमेशा लाइव एटिन्यूएटेड जो रहता है वो सबसे ज्यादा इम्यून रिस्पॉन्स ट्रिगर करेगा. इसके लिए कोई बूस्टर डोज की जरूरत नहीं पड़ेगी, क्योंकि वी आर ऑलरेडी टेकिंग दी वीकेंड पैथोजन, लाइक वीकेंड एंटीजन हम ले रहे हैं. So these are the things associated with the immune response to different types of vaccines. Now, next part. So before studying this, let's solve some question. Which one of the following vaccine types provides the most robust and long-lasting immune response, often without the need for boosters? Now answer me quickly. कौन सा वैक्सीन टाइप जो है लॉन्ग लास्टिंग इम्यून रिस्पॉन्स देगा विदाउट द नीड फॉर बूस्टर डोज? आई एम नॉट गेटिंग अ सिंगल आंसर. ओके, एक आंसर आया.
लाइव एटेन्यूएटेड.
ओके, राइट, लाइव एटिन्यूएटेड. सो यू शुड ऑलवेज रिस्पॉन्ड. अगर मैं इतने एफर्ट्स लगा के रात के 9 बजे भी पढ़ा रही हूँ, सो यू शुड ऑल्सो आंसर मी. गलत हो तो क्या है, सबको सब कुछ नहीं आता और एक बार में समझ नहीं आता है, तो उस चीज की चिंता मत करो कि मुझे गलत आंसर है. माइक अनम्यूट करो और जो भी है डाउट उसको पूछो, पूछना चाहिए हमेशा. सो अभी जस्ट जो मैंने पढ़ाया वो ही क्वेश्चन है ये, कि कौन से वैक्सीन में द इम्यून रिस्पॉन्स इज अप टू अ वेरी लॉन्गर ड्यूरेशन एंड यू डोंट नीड अ बूस्टर डोज. So it is nothing but the live attenuated. We are taking the live antigen. So इसके लिए हमें जरूरत नहीं पड़ेगी डबल डोज की. और ये क्या करेगा, ये CMI और ह्यूमोरल रिस्पॉन्स दोनों को ही एक्टिवेट करेगा. Now next
And
question.
strong antigen means what? Whether it is concentration of antigen or viral load. If it is the more viral load, then the immune response will be more. What does it mean exactly, strong antigen?
Both are appropriate. If we are saying strong, let's say a weak strain is there. But if that weak strain is there, but if you count the CFU, like we count in bacteria, so if the weak strain population is high, then it will impact a lot. Then you can say that this is a weak strain, but its population is high, then it can lead to a very, very high immunological response. But if we are saying that this is a strong or a very, very potent viral, it means it's only a single colony will be able to cause a lot of harm in your body. So both the things are true in that case. Weak strain, more population will cause you a lot of harm. And if it is a very potent one, its less concentration will also be equally harmful.
Okay, got it. Thank you.
Thank you. Now, next question. Which immune cells are primarily responsible for producing antibodies after the vaccination? Macrophages, T helper cell, plasma B cell, TC cell.
Plasma B cells.
B
Yes,
cells.
right. Plasma B cells. So plasma B cells are differentiated B cell which are rapidly synthesizing the different kind of antibodies. Which component is commonly added to subunit vaccines to enhance the immunological response? Antibiotic, booster dose, adjuvants, toxoid.
Adjuvants.
Yes, adjuvants. We add adjuvant to increase the immunological response. Now आपको ये भी पता होना चाहिए कि कौन कौन से mechanism से. So we have already studied those mechanism. mRNA vaccines work by delivering preformed antibodies, inserting viral DNA into host cells, stimulating antibody production through inactivated virus, and coding antigen that are translated into protein in host cells.
D.
Encoding the antigens that are translated into
Yes,
Encoding
absolutely
antigens.
correct. Yes, correct. Very good. mRNA वैक्सीन इट इज नेम्ड एज mRNA. अगर mRNA है तो वो क्या करेगा? वो ट्रांसलेशन करेगा. ऑलरेडी ट्रांसक्रिप्शन होके mRNA बन चुका है. नाउ द नेक्स्ट स्टेप विल बी ट्रांसलेशन. सो वो mRNA क्या एनकोड करता है? इट हैज द antigen gene that is translated into antigenic protein inside the host cell. So mRNA vaccine का यही principle होता है कि वो antigen का gene encode करती है and it is translated inside the host body. Now back to the topic. So we have covered up till quickly revision. So we have covered the mechanism of vaccine induced immunity and what are the different kind of immune response to different types of vaccine. Now we will be focusing on this duration and memory of immune response, how long it can be there. So see, this is the important part, and you have also have observed this many a times in your report. So whenever we are having any kind of viral infection, or in case of COVID-19 also, we are having this report kind of thing, like your doctor will prescribe you to measure the antibody level. Antibody levels are measured like the IgM and IgG levels are always monitored. Like which of the following are released and what are the exact time at which they are releasing. So memory B cell and memory T cell ensures rapid secondary response upon re-exposure. So as previously I was talking about, if you are having vaccine 1 and vaccine 2, like these different kind of vaccine for a particular antigen, let's suppose for COVID only. So for COVID-19, we are having vaccine 1 and vaccine 2. So how you will say that V1 is better or V2 is better? So if you have to give recommendation to a patient that kaun sa lagaun main, so how you will recommend them on the basis of the effectiveness, on the basis of how much immunological memory it is inducing. So memory B cells and memory T cells are measured whenever the vaccines are administered after a certain period of time, like after a chronic exposure to the vaccines. These things are measured, that which vaccine has induced a lot of memory B cell and a lot of memory T cell. As we have just studied that Th1 and Th2. So Th1 was doing the stimulation of CMI, cell mediated immunity, and it is triggering the humoral. So Th1 is associated with the CMI, and also one immunological memory T cell was saved for future use, and in this one immunological B cell was stored for future use. तो ये जो दो यहाँ पे बच गए, ऐसे करके कितने मल्टीपल रिएक्शंस बॉडी में चल रहे होंगे. सो ऑलवेज द टाइटर इज मेजर्ड, लाइक कितना कौन से वैक्सीन ने कितना मेमोरी बी सेल और मेमोरी टी सेल दिया है. So live vaccines often induce longer memory, years to lifetime. So whenever you have taken the live attenuated one, no such booster dose is required, and you will be protected for lifelong, because they are having the live antigen, and that will uh, produce you a lot of memory B cell and memory T cell for lifelong. So whenever this same antigen will your body will encounter the same antigen for the second time, quickly your memory B cell and memory T cell will be activated. At that time, no such new things will be made, but what we will use, what our body will use previously stored memory B cell. Like whenever you are preparing notes for a particular exam, let's say exam XYZ, so if you have already prepared the notes of the required unit, so for the second time you will not again make the notes. फिर से नोट्स बनाने में टाइम वेस्ट नहीं करोगे. व्हाट यू विल डू, यू विल क्विकली रिवाइज. सो हमारे बॉडी में भी सेम चीज होती है. क्यों वेस्ट करना है अपना एनर्जी सारा फिर पूरा स्क्रैच से शुरू करने में. अवर बॉडी क्विकली एनालाइज दैट ये तो पहले भी आया था, we have encountered this antigen previously, so we have to just what we have to do, we have to just hint the memory B cell and memory T cell कि फिर से antigen आया है, we have to kill it out. So live vaccine में we are having a lot of memory B cell and memory T cell, but in killed subunit vaccine they may require booster dose because the antigen is not that potent. So memory depends on, of course, the vaccine type, of course the host immune status. You cannot say that the immunological the immunocompromised people will have a high titer of memory B cell and memory T cell. Despite giving them the live vaccine, it depends on the status of their immunity, because in the immunocompromised people, they have a very, very low amount of the memory B and memory T cell. So it depends on the host immune status. So if you are a immunocompromised people or a people with some disease history, so you cannot say that you will be having same amount of the immunological memory as a healthy individual. No. Also the use of adjuvant. Some adjuvants are very, very helpful for increasing the immunological response. So if the immunological response is increased, memory B cell and memory T cells will be increased. So it depends on adjuvant 1 to adjuvant 2. Like adjuvant 1 will be capable of triggering our immune response a lot. Suppose A2 is not that much effective as A1. So it is also a factor which will influence that whether the memory B and T cell will be made or not. And we cannot forget about the role of the route of administration. So see, everything is linked with each other. This is known as coherence. So this was the unit 6. In unit 6 we are studying all these things, but we know about the route of administration. यानी हमें route of administration का भी पूरा theory पता है कि किस किस route से vaccines दिया जाता है. So always study like this way. यानी पहले jump करके advanced level में नहीं जाना है, पहले basic ground level की चीजें पढ़नी है, फिर आगे जाना है. So we are having different kind of like factors which will decide that whether the immune response will be for a longer period of time. Longer कब होगा? जब memory B cell और memory T cell will be in a substantial increase जब होगा तभी होगा वो longer time के लिए. If it is for only अगर आप सब यूनिट वैक्सीन की बात करें, किल्ड की बात करें, so it will not for a lifelong time, because of course the antigen is not that potent. So this is a vaccine, how vaccine is responsible for eradicating out any of the antigens. So vaccine provides defense against bacterial or viral pathogen by inducing the immunological memory. So in today's lecture, I just told you about the that vaccines effectiveness is measured in terms of immunological memory. If it is able to provide the patient with memory B as well as T cell, then you can say that this vaccine is very much effective. You can give it to the patient. So whenever this graph is related to it is time dependent, and this is our adaptive immune response. So whenever you are saying that after a certain period of time of taking vaccine, your antibodies level will be monitored, like it will be monitored the adaptive immune response. So after a very long period of time, it will be monitored. Like initial exposure of antigen, if the antigen is initially entered in your body, then your body will take some time to analyze that something is happening inside the body. Some antigen is already there in our body, so it will uh, alert all the immune cell कि हाँ कुछ disturbance body में हुआ है, body का जो homeostasis है वो क्या हुआ है, it is disturbed. It means there are some antigen which already our first line of defense failed to eradicate them, and consequently they have entered in our body. So जब वो recognize कर लेते हैं कि हाँ कुछ आ गया है, अब हमें activate होना है, तो वो response generate करते हैं. So you can see एकदम बेसलाइन को टच कर रहे हैं, यानी एकदम जीरो होता है जब वो एक्सपोजर इनिशियल हो रहा है तब. बट वंस इट इज रिकॉग्नाइज्ड बाय एपीसीज, वो क्विकली क्या करता है, वो इम्यूनोलॉजिकल रिस्पॉन्स जनरेट करता है. बट यू कैन सी You can see the what to say intensity. ये फर्स्ट टाइम का है, जब ये फर्स्ट टाइम आ रहा है तब कितना कम है, बट इफ यू आर मेजरिंग इट आफ्टर द सेकेंड और द सब्सिक्वेंट एक्सपोजर, यू कैन सी, if you can say this IR1 and you can say this IR2, so IR1 is very much very lower than IR2, यानी IR2 इम्यूनोलॉजिकल रिस्पॉन्स 2 बहुत ज्यादा है. सो पहला जब आता है उसे कहते हैं प्राइमरी, दूसरा जो आता है उसे कहते हैं सेकेंडरी. सो सेकेंडरी इम्यून रिस्पॉन्स विल ऑलवेज बी हायर. इस क्वेश्चंस कैसे बनेंगे सी सेक्शन में, CSIR कैसे क्वेश्चन देगा, इसी टाइप का कुछ ग्राफ दे देगा एंड यहाँ लिख देगा वैक्सीन, मतलब डबल ग्राफ कुछ दे देगा और लिखेगा वैक्सीन 1 का जो सेकेंडरी इम्यून रिस्पॉन्स है वो लेट से एंटीबॉडी टाइटर कुछ देगा 4:1 है, एंड वैक्सीन 2 का 2:1 है. तो कौन ज्यादा इफेक्टिव वैक्सीन है? ऑफ कोर्स जिसका सेकेंडरी रिस्पॉन्स ज्यादा होगा, इट मीन्स उसका इम्यूनोलॉजिकल मेमोरी ज्यादा है, इट ऑलरेडी रिटेन द प्रीवियस इम्यूनोलॉजिकल एक्सपोजर ऑफ द एंटीजन, वॉट एवर इट इज, बैक्टीरिया हो वायरस हो, डजेंट मैटर. तो जिसका जिसका इम्यूनोलॉजिकल रिस्पॉन्स यानी कि एंटीबॉडीज का टाइटर ज्यादा होगा, वो ज्यादा इफेक्टिव वैक्सीन होगा. सो दिस ग्राफ इज इंपॉर्टेंट. एज पर टाइम जब भी ये सेकेंड एक्सपोजर होगा इस एंटीजन का, what it will do, the response will be faster and the response will be slower. यहाँ पे स्लो होगा और यहाँ पे फास्ट होगा, बिकॉज वो प्रीवियसली आ चुका है हमारी बॉडी में, मेमोरी टी सेल मेमोरी बी सेल ऑलरेडी है, तो स्क्रैच से शुरुआत नहीं होंगी चीजें. सो क्विकली रिस्पॉन्स जनरेट होगा एंड दिस इज नोन एज द सेकेंडरी इम्यून रिस्पॉन्स. And you can say that secondary, in primary we are always having IgM. IgM is produced first, followed by the class switch to IgG and IgA. यानी कभी भी आपके बॉडी में अगर IgM का लेवल ज्यादा आता है एज कंपेयर टू IgG, इट मीन्स कि इनिशियल एक्सपोजर हुआ है वायरस, मतलब जो भी एंटीजन है उसका, it is only, you can say that, you can interpret it that it is the infection is in only primary stage. If the levels of IgG is more, it means कि infection जो है वो severe हो चुका है. So in primary immune response, generally it takes time about 7 days, and also it depends on person to person, ये depend करेगा, because these factor will be guiding the overall output, host immune status, and also the actually every 4 factor will be influencing the output, but majorly host immune status will decide that whether it will take 7 days, 4 days, 3 days, or like that. So IgM will be produced first, followed by class switch to IgG or IgA, secretory IgA. Memory B and T cells are generated. Secondary immune response, rapid and robust due to memory cell, higher affinity antibodies dominated by IgG or IgA. So you can say that live attenuated, the duration will be, this is the duration of immunological response, lifelong after 1 to 2 doses. Inactivated subunit, few years, needs booster dose. mRNA, month to years, depending on the boosting. So this is the immunological response related to different kind of vaccine. So you have to remember कि कब first line of defense activate होता है, कब primary generate होता है, कब secondary generate होता है, and also you can say that these factors are going to influence the overall thing, like vaccine formula factor influencing the duration, vaccine ka formulation and the delivery route, use of adjuvant, host factor like age, because older age people will always having low immunity, nutrition, what type of nutrition you are taking, immunocompetence means if you are a immunocompromised people, or if you are a person who are having a very highly active immune response like hypersensitivity kind of people, यानी एकदम ज्यादा hyper active भी रहेगा immune response, तो क्या बोलते हैं उसको, हाइपर सेंसिटिविटी. यानी कुछ लोग एलर्जिक होते हैं, जैसे ही डस्ट में जाएंगे तुरंत एलर्जिक रिस्पॉन्सेस जनरेट होंगे, तो हाईली कॉम्पिटेंट भी होने से प्रॉब्लम है एंड एकदम स्लो पेस में होने से भी प्रॉब्लम है. एंटीजन परसिस्टेंस एंड मेमोरी सेल सर्वाइवल. सेम ही एंटीजन बार बार आ रहा है तो भी अलग रिस्पॉन्स आएगा. हमारा जो इम्यूनोलॉजिकल रिस्पॉन्स है वो एकदम यू कैन से स्टक हो जाएगा. दिस इज नोन एज एंटीजन परसिस्टेंस. Site of memory cell. Memory B cells circulate in blood as well as in lymphoid organs. Central memory T cell is only in lymphoid tissues. Effector memory T cell is in peripheral tissue like lungs and our gut. इसीलिए बोलते हैं कि gut health को